Hello, everyone. My name is Aaron Pomerantz, Director of Global Segment Marketing at Oxford Nanopore Technologies, and it's my pleasure to welcome you all to our webinar today. I'm delighted to be joined by the team at Ascend Advanced Therapies, who will share how they're applying Oxford Nanopore sequencing to enable faster, high-resolution insights in biopharmaceutical quality and development. After their talk, the team will be available to take your questions in a live Q&A, so please add your questions into the Q&A tab, which you can find to the right of your screen. You can also upvote the questions you would most like answered, and we'll try to get through as many as we can. This webinar is being recorded and will be made available on demand after the broadcast. Before I hand over to our fantastic speakers, I'd like to first kick off with a little background on this technology and how we see it redefining standards in advanced therapeutics today. As many of you know, the landscape of drug development has changed dramatically over the years. As biologically derived therapeutic modalities emerge and evolve, they're trending towards larger, more complex, and more challenging to characterize. On the left, you have traditional small molecules like aspirin, chemically simple, with well-defined structures and synthesis reactions. But as we move towards biologics such as small and large proteins, viral vector platforms, and cell-based therapies, the complexity increases dramatically. With this increase in complexity comes a corresponding rise in the range of critical quality attributes that must be monitored. These products often show higher variability and a greater risk of impurities, which can have significant implications for safety and efficacy. This means there's now a greater need for advanced analytical methods, tools that can provide high-resolution insights into molecular structure, genome integrity, and overall product quality, not just during discovery, but throughout process development and manufacturing. And that's where Oxford Nanopore Technologies comes in. We've developed a new generation of single molecule sensing technology, a platform built to deliver the kind of advanced analytics that today's therapies demand. Our sequencing instruments can scale to any lab need, from benchtop experiments to large-scale production environments, all using the same underlying technology. Here's how it works in a nutshell. Strands of DNA or RNA are passed through tiny protein nanopores, and as the molecule passes through, changes in electrical signal are measured and translated into the sequence of nucleic acids. Because nanopore sequencing can read native molecules directly, it allows for virtually unrestricted read lengths and the ability to detect base modifications alongside sequence data. That means you can capture comprehensive DNA, RNA, and epigenetic information in a single platform, giving you richer biological insights without the limitations of traditional sequencing technologies such as short read lengths, PCR biases, and high instrument costs. The need for comprehensive sequencing isn't just theoretical. When it comes to high-value biological materials, what you're missing can truly matter. Hidden errors in samples can jeopardize entire projects, compromise reproducibility, and delay time to market, often without users even knowing they're there until it's too late. A recent study from Bai et al. Analyzed thousands of lab-made plasmids and found that nearly half contained undetected sequence or design errors. To me, studies like these should really be a wake-up call for anyone in this field, as these are the very reagents used to express your genes of interest, produce your viral vectors, and ultimately advance your pipeline. The antibody field has also recognized quality and reproducibility issues for many years now, which is likely exacerbated by a lack of routine sequencing. As researcher Kathleen Zaglinski puts it, sequence-based validation of antibody reagents can be a valuable way to check what you're using is actually what you think it is. This is where comprehensive profiling with Oxford Nanopore makes a difference, giving you complete visibility into the constructs, the vectors, the biological reagents that you depend on, so you can spot problems early before they cost you time and resource. Because advanced therapies such as gene, RNA, and cell therapies are biologically based, variability and errors can appear at multiple points along the development and manufacturing process. Comprehensive sequencing therefore plays a critical role as an analytical checkpoint at nearly every stage, and can help you confidently answer questions like, is my critical plasmid raw material free from sequence errors that could impact downstream yield or fidelity? Are my vector genomes intact and accurate, free from truncations or impurities that could impact potency or safety? Is my biological product fully profiled for identity, integrity, and purity before moving to scale up? Did I deliver the appropriate edit or response at the intended target site, And were there any off-target effects? For example, an unexpected integration event that could cause potential oncogene activation? And is my final product fully characterized across critical quality attributes to de-risk regulatory submission and accelerate approvals? So how can Oxford Nanopore help address all these critical questions? Let's take a look at a few use cases. Plasmids are foundational raw input materials for many advanced therapy workflows, serving as templates for viral vectors, RNA therapeutics, and engineered cell constructs. But if a plasmid carries even a small error, such as a deletion, an inversion, a contaminating plasmid, it can lead to wasted time troubleshooting downstream issues. With Oxford Nanopore sequencing, you can quickly verify complete plasmid constructs without having to design or order any primers to confirm that the construct you're working with is actually what you expect it to be. The data shown here demonstrates complete coverage and assembly into a single annotated plasmid map. As users like Dr. Stephanie Maiden nicely put it, at least twice in my career, this could have saved me from spending months on faulty plasmids received from others. Always sequence your stuff. Now let's look at an example with AAV genome analysis, which really highlights the limitations of traditional short-read sequencing platforms. On the left, you can see a coverage plot generated from short reads, and you'll likely be able to spot regions with sharp drops in coverage, especially high GC-rich regions and in the inverted terminal repeats, the ITRs. These are critical elements in AAV vectors, but because of the inherent biases and limitations in short-read sequencing, you're effectively blind to potential issues that could affect the integrity and safety of the gene therapy product. On the right, we can see why Oxford Nanopore sequencing is uniquely suited for characterizing these viral vectors, delivering complete, uniform coverage across the AAV genome from ITR to ITR. This allows for direct detection of not only full-length vectors, but can also spot if there are any truncated vector genomes, chimeric sequences, and residual host or plasmid DNA impurities, providing a more accurate view of what's actually inside your vector prep. And this type of platform isn't just useful for AAV. It's equally powerful for other gene therapy systems like lentiviral vectors. Oxford Nanopore's full-length direct RNA sequencing provides a major advantage. Unlike other sequencing technologies, which rely on converting RNA to cDNA and introduce PCR bias, we can sequence entire RNA molecules directly. This means you can detect all transcript species, such as full-length, spliced, or truncated forms, providing an unbiased complete picture of the lentiviral RNA population. Lastly, let's take a look how this applies to mRNA vaccines and therapeutics. Today, verifying mRNA CQAs requires multiple separate assays, typically run on different instruments by different operators, each measuring just one attribute of the molecule. Oxford Nanopore is setting a new standard in mRNA quality, again as the only technology on the market that can directly sequence native RNA molecules. That means you can capture the molecule exactly as it is, including base modifications such as N1-methylpseudouridine, poly-A tail length estimation, and transcript completeness. This provides a truly multi-attribute test, one platform that simultaneously measures identity, integrity, and purity of your mRNA product, as well as its plasmid precursor. The examples we've just seen highlight a bigger picture. What if a single platform could accelerate discovery, improving product understanding, and reduce risk all the way from R&D through manufacturing? That's really the vision behind this platform, providing one system that can deliver value across every stage of the biopharma pipeline. In biomarker and target discovery, nanopore sequencing enables rich multi-omic data, capturing DNA, RNA, and even epigenetic modifications, to accelerate the identification of novel biomarkers and therapeutic targets. The same platform can then verify engineered plasmids, synthetic DNA, or RNA constructs, ensuring that design accuracy and fidelity are confirmed before moving downstream. During vector and cell line characterization, we can achieve full end-to-end genome integrity assessment, helping confirm precision, stability, and product consistency. And finally, in biomanufacturing, our regulated devices and tests support deep profiling of critical quality attributes, reducing regulatory risk and helping accelerate time to market. To wrap up, we've seen how Oxford Nanopore delivers a differentiated platform, one that's helping redefine analytical standards across the biopharma industry. First, it provides richer, more actionable insights, capturing single molecule, multi-omic data that reveals not only sequence information, but also structural variants, base modifications, and methylation patterns, all from a single workflow. Second, it enables faster time to development. With near-sample, real-time sequencing, users can access results in hours instead of days or weeks, accelerating decision-making and reducing costly delays. And third is a scalable and regulatory-ready platform, from benchtop devices for early research through high-throughput instruments suited for process development and GMP environments. That flexibility means the same technology can support you across every stage of discovery, development, and manufacturing. That's why we're really proud to see CDMOs like Ascend Advanced Therapies leveraging this technology to give their partners deeper, faster insights into the quality of today's most advanced biological medicines. So with that, I'd like to hand it over to Rebecca Feiner, Manager, Technology Development at Ascend, to share their excellent work. Over to you, Rebecca.
Welcome also from our end to today's webinar. We're quite excited that we have the opportunity from Aaron and Oxford Nanopore Technologies to present our journey into AAV characterization using NGS today. Our whole journey started in 2015, and at that time our Munich site was still part of the product development company Freeline Therapeutics. Freeline at that time had clinical trials ongoing for a factor IX therapy, and the AAVs used in these clinical trials have been produced in HEC293 T cells in an adherent iCELLis system, and this delivered benchmark quality at that time. However, the FDA pushed back on the use of the 293 T cells due to the oncogenic potential of the large T. The problem was not solved solely with our internal analytical capabilities, so we needed a partner. And in 2017, we started to establish a pipeline with an external partner. Our NGS data, together with transcriptional profiling assays, nested PCR approaches, as well as, and in the end, the NGS, helped to de-risk the use of the cell line by delivering data on all packaged DNA impurities, which included plasmid as well as host cell-derived sequences. In 2019, we finally got the go from the FDA to be able to use the cell line as well as the products for the clinical trials. We actually at that time saw the great benefit of NGS for AAV characterization, and this is why we started in 2020 to develop our own internal NGS pipeline. And we used this pipeline to characterize AAVs in development stages, and because we were optimizing our manufacturing and plasmid systems. We also used the data, we used the pipeline to generate data for toxin engineering batches as well as for GMP material. In 2023, our Munich site that is specialized on CMC development was acquired by Ascend Advanced Therapies. And overnight, our focus changed from being a product development company to being a CDMO. We still continued development of processes on NGS and used NGS for the characterization of our AAVs. But in addition to that, we were now also able to offer all our analytical assets to clients. We still, as I've mentioned, developed our NGS platforms as well and refined our pipelines. And this development, or part of this development, was in the beginning of this year published in our first peer-reviewed publication. Meanwhile, the gene therapy landscape has changed quite a lot. Regulatory requirements have become more stringent and more hurdles had to be cleared. And with that, the acceptance of NGS as a valuable tool for analytical AAV characterization has grown. Back in 2017, NGS methods were still relatively unknown in the AAV gene therapy field, and furthermore also pretty expensive. In recent years, however, costs have decreased and the ability to process large data sets in shorter time frames has improved considerably. As a result, the perception of NGS has shifted. Over the past 5 years, we've seen an increase of NGS data presented in publications and at conferences. And this clearly shows that NGS has moved beyond just being an R&D tool. It's becoming a standard requirement into our field and is even encouraged for usage in a regulated GXP environment. NGS hereby supports the analysis of several critical quality attributes of AAVs, including genome integrity, truncated vector genomes, residual DNA impurities, as well as fusion sequences. The confidence and trusting data from the powerful NGS platforms came also through other methods that confirmed results. One powerful method standardly used in the AAV characterization is digital PCR. This method is superior when it comes to absolute quantification of genomes and DNA impurities. However, ddPCR methods lack the full picture of all packaged sequences because you can only analyze the amplicon that you have designed prior. In other words, it gives you a really precise answer, but only to a very specific question. In the bottom right image, you can see this as an example for 2 known impurities. But what about everything that is in the middle and unknown? This is where NGS comes in. It provides a comprehensive overview on all package sequences without the need for any prior sequence information. And the bottom left image illustrates this broader insights. So you are now actually able to see what happens in between our amplicons. And this is why we see as an orthogonal method. It doesn't replace other methods like the digital PCR. It complements them. Coming back to the beginning, we had to make a decision. The term next-generation sequencing is used for a group of technologies that can be divided into short-read technologies as well as long-read technologies. Early work in the AAV field focused on short-read technologies, and in 2015, a couple of publications came out that showed a high accuracy and deep sequencing coverage, which led to really good results, especially for the analysis of vector identity. However, we've seen challenges around the resolution for DNA sequences with a strong secondary structure as well as a high GC content, which is especially problematic for the inverted terminal repeat structures of the AAV genome. Long-read methods that have the advantage of covering a full AAV genome in one read were at the time in 2015 more cost-intensive and also had a much lower read depth. Most of the challenges of long-read technologies were overcome by nanopore and smart sequencing in the recent years, and in addition, they were also able to resolve the challenging RTRs. All of this is the reason why in the AAV field a shift has been observed from short-read technologies to long-read technologies in the past 5 years. We early on decided to explore the use of nanopore sequencing due to the advantages of the low sample consumption, it being a relatively cheap and easy to implement method, and the opportunity to cover full transgenes. In the beginning of our journey, when we started working with nanopore sequencing, actually the most issues we've observed were with the per base quality of our nanopore NGS reads, which were a lot lower in comparison to PacBio or even Illumina sequencing. And this is why other people smiled at us for using NGS. To give you a bit of an overview on what the data looked like, on the left-hand side, you can see a graph that shows shows a summary of sequencing results from the same AAV batch that we have sequenced over the years with the technologies that were available at that time. So in 2017, when we started, the V9 chemistry as well as the Guppy base caller were the method of choice. With that and our general AAV batch, we were able to get a per base quality over Q15, which was not as good as of the other methods. In 2023, Oxford Nanopore introduced a new chemistry V14, a new flow cell type, and the Dorado base caller. And all of these changes improved the quality significantly above the Q20 score. As of today, in 2023, the per base quality reaches comparable levels to the other two methods, showing the great potential of this method. So by just introducing a new base calling model, we can now exceed the Q30 score. Before now handing over to my colleagues Kati and Flo, I would like to go back to the beginning of our journey. In the beginning, in 2017, we were by all means experts in the NGS field. We were experts in the AAV field, and we had specific questions that we wanted to have addressed, and we wanted to use NGS for it. However, initially we were super overwhelmed by all the data that was generated and also by the new terms that have been used by the NGS experts. We worked together with an external partner, and we actually struggled to interpret their results sometimes. And also, we got a lot of results, but we had specific questions. So we actually had difficulties to get exactly the data that we needed to answer those questions. We have come a long way, and now we would consider ourselves AAV experts with a really strong NGS background because we have learned our lessons. We are now able to write our own bioinformatic pipelines, we have developed our own wet lab processes, and we do understand where people do still struggle with today. And all of this leads to the offer that we offer clients. We do understand that not everyone is using the same manufacturing platform and the same plasmid system. People do use one plasmid, two plasmid, three plasmid systems, and all the bioinformatics needs to be adapted to that. We offer this adaptability and flexibility of our pipeline because we come from the same background. We have always used different plasmid systems and evaluated different systems at different stages, so not every offered method from external providers delivered the results that we were actually looking for. We also do offer a deep data dive whenever we see something that is a little bit up, that needs a little investigation. We go usually beyond the standard, and this is what we think differentiates us from others, that we deliver an expert interpretation that brings together the NGS as well as the AAV world and helps to interpret the data. But I will stop here because our focus today should be on actual data and science, and also on the latest approaches that we have implemented into our pipelines. And with this, I will hand over to my colleague Katrin, who's one of our experts for wet lab and dry lab activities.
Yeah, thanks Rebecca for the introduction. My part in this webinar focuses on the first part of the data analysis. So first of all, AAV is produced by co-transfecting mammalian cells with plasmids encoding the gene of interest, AAV replication and capsid proteins, and helper functions. And this process can introduce impurities during production, and this is what I want you to show here. What do we actually get if we analyze our AAV batch? So while in an ideal world, only the vector cassette would be packaged into our AAV, and only this would be analyzed via NGS. However, we all know that reality is different. We do have the full AAV vector cassette as the main component, but also know that the nature isn't perfect, and also partials are packaged into our batch. So we find transgene fragments with only one ITR or even without any. Then there's the vector plasmid backbone that can appear in the manufactured AAV batch, as well as the helper plasmid or parts of it, and of course the manufacturing cell line DNA. And all of these impurities have different origins and occur at different levels depending on the manufacturing process. Additionally, all packaged impurities can potentially remain transcriptional active or may lead to unintended protein expression, and this is exactly why deep characterization of our AAV batch is so important. So sequencing AAVs first comes with the task to prepare suitable libraries for sequencing. The AAVs have a single-strand DNA genome, which needs to be first extracted and then converted into double-strand DNA to be amendable for library preparation and subsequent sequencing. So we put a lot of effort in this pre-library step and compared different methods to generate our double-strand DNA template. So we have the enzymatic conversion and annealing. The conversion step should theoretically convert all single-strand DNA into double-strand DNA regardless of the properties, but it maintains the length of the original fragment. So annealing, on the other hand, just works with temperature, as the AAV genome is present in plus and minus strands that can combine to a double-strand DNA. We initially assumed that annealing would work fine for the abundant vector genomes, but it might struggle with rare impurities that don't easily find a matching strand. And to this end, we invested effort in enzyme-mediated second-strand conversion. And this work was described in detail in our publication. But concluding, we can use both, and we do use both methods based on the question that we want to address with the experiment or the intent of a given NGS study by a customer. To decrease the likelihood of recombination competent AAVs that appear through recombination events during production, we have developed our proprietary EPIC platform that is a two plasmid split system. In a triple transfection plasmid system, rAAV and cap genes are sitting on one plasmid, and then we have the vector plasmid and the helper plasmid. In our approach, rAAV and cap genes are divided onto two different plasmids. The first plasmid contains adenoviral helper genes, alongside rAAV genes, which is essential for replication and packaging. The second plasmid contains our gene of interest alongside the cap gene for the essential proteins VP1, 2, and 3. And these two plasmids are then transiently transfected in HEC293 cells. And I want to show you here our developmental AAV9 batch from a 50-liter bioreactor. After sequencing, when we look at the size distribution of our reads, we detect the size of our transgene, which is approximately 3 kb in size, as well as a lot of shorter reads here, where Flo will tell you a little bit more after my part of the presentation. And the plot here shows the overall coverage in gray and the schematic transgene above it, with the ITRs on both ends, the promoter region as well as the transgene and poly A, that is all nicely covered over the whole sequence with no truncation hotspots. The coverage, however, slightly drops towards the end of the ITRs, which might be due to the complexity of the challenging secondary structures by the ITR. But more important is this pie chart here, where we see that over 96% of reads map to the transgene, whereas only 2.34% are transgene plasmid backbone impurities, 0.71% are haplo-plasmid impurities, and only 0.04% are host cell-derived impurities. And these impurity data were confirmed data from orthogonal methods like the ddPCR. And in the next slide, I want to highlight the impurity data a little bit more. So having a closer look at the plasmid-derived impurities, we are unable to uncover the starting points of these impurities. And the transgene plasmid backbone impurities as well as the haplo-plasmid impurities show a P5 promoter-mediated reverse packaging. In the upper graph, we also see ITR reverse packaging or bidirectional packaging, and in the haplo-plasmid graph, we only detect the P5-mediated reverse packaging in this area. Both graphs are, just to zoom in, they show very low levels of impurity and are not quantitative. And this is for better understanding the packaged DNA impurities and to uncover the starting points. And also with these NGS data, we see so much more as with, for example, the ddPCR, as Rebecca in her introduction slides already explained. So it depends where the primers for ddPCR are located to estimate how much impurities we really have. So here in the cap gene, for example, we would miss a big fraction of impurities if we would place the primers here in the N-terminus. And vice versa, we would overestimate impurities by placing primers in this area here. And with NGS and those coverage plots, we can definitely make a better statement about the transcriptional potential. And then we have the host cell-derived impurity that is of special interest for regulatory authorities because of its potential oncogenicity and immunogenicity. The longer the fragment, the higher the likelihood of critical safety implications. And in our data, the majority of host cell-derived DNA impurities are smaller than 500 nucleotides in size and make up only less than 0.1% of all the reads. The quality of nanopore sequencing data increased so much the last year. So however, we wanted to compare our sequencing data with another long-read sequencing method, namely PacBio Smart sequencing. And we sequenced the same AAV9 batch externally with a commercially available sequencing provider. And annealing was here the standard protocol. And to directly compare it, we also annealed our samples and loaded it on our nanopore device. And looking at the coverage plots, we see the same coverage distribution for both long-read technologies, but generally can make the same statement, which is good. Smart sequencing is cleaner with less errors and also show the same trends than the nanopore data do. In those coverage plots here, each gray horizontal bar below the purple line corresponds to a single sequencing read that has been aligned to the reference genome and shows coverage as well as any mismatches or variants. The strongest difference, however, is the statistics. So we see around 95% transgene mapping for our nanopore sample, but only 90% with smart sequencing. Additionally, the plasmid backbone impurity for smart sequencing shows a twofold increase than our nanopore data, and hapoplasmid and host cell-derived impurities are comparable. However, the data from orthogonal methods support and agree more with the lower values of the nanopore impurity data. So we suggest that this might be a lab process derived difference. And overall, nanopore sequencing provides comparable results at lower cost and material consumption. And all the data I just showed you are part of our publication, where you'll get more information and dive deeper if you want to. But if there are any questions, just drop us a mail for clarification. And then I hand over to Flo, who is explaining our data analysis more from a bioinformatics side, and he's in charge of all the bioinformatics as well as wet lab and dry lab processes in our NGS team.
Yeah, thank you, Kati, for this kind introduction and also for highlighting the amount and the wealth of data that we can actually get out of nanopore sequencing and how much we can actually learn about our rAAV batch. However, there was always one thing that kind of puzzled me and also my colleagues. And this was that we never saw that one sequence is really one sequencing read. So if you see here, we have a library and we load it on a tape station, which is basically a modern way of doing an agarose gel. We see some shorter reads, but mainly we see, as expected, a dominant peak at the size of our full-length genome. If we sequence this library, however, we always saw many more small reads than expected. And you saw also real data before by Kati. We could never see really the distribution that is in our tape station. And if you think about something like this and you want to solve what is maybe happening, it's always a good idea to really take a step back and think about the method once again. How is actually this method working? So we have a three-dimensional flow cell, and in this flow cell we have shorter or longer DNA pieces with a motor protein that are looking for a pore, and only those that are attached to a pore will get sequenced. And we know that during a run, the flow cell is exhausted much faster than all DNA pieces to be sequenced. This we know because we can recover the library and load it on a fresh flow cell, and we get a lot of new information. So it means only a fraction of the reads gets really sequenced. And which reads are sequenced is largely dependent on how fast they are actually moving, so on their diffusion speed. And this, of course, is dependent on their size. So this means next to unpredictable and not reproducible variations in the sequencing output from run to run, from flow cell to flow cell, we also have a size-dependent effect that is predictable and reproducible. We see it always the same. And if we have these two things, that something is predictable and reproducible, it becomes correctable. So how can we correct now our size-dependent effect? We need a marker with varying sizes that are present in equimolar amounts. So how can we get this? Well, we can simply take a lambda phage DNA and digest it with a restriction enzyme. And we bought this actually to have it in a very reproducible way, but you can also do it by yourself and simply digest a lambda phage DNA. And you see here, if you load this on a tape station, we have DNA fragments of different sizes that are all present in equimolar amounts. They are actually, tape station is measuring the amount of something, so the mass, and of course the larger the fragment gets, the more mass it has, and the more dye intercalates. But they are all present in the same molar amount. If we sequence this with Oxford Nanopore, we see a strong selection of short fragments. So we have a lot of them, and the larger the fragment gets, the less likely it is to be sequenced. And we can take these data now to plot a function, and you see here it is a non-linear function, and you can basically do a regression now and make an exponential fit. And this fitting function we can then use to correct our data. An important note is that this function is not linear, means it somehow confirms that we really have this size-dependent effect due to the differences in diffusion. Because after the Stokes-Einstein law, the diffusion speed is inversely proportional to the particle size or the hydrodynamic radius, which in our case is more or less the DNA length. So how does it look now with real data? If we take a library quality data of a really great library, we have a very dominant full-length peak and some shorter fragments, we can really now bridge the gap between these quality data and our NGS data. Because the raw data that comes out of the run shows the dominant full-length peak, but also many more short fragments shorter than 500 nucleotides than expected. If we correct these data now with a co-sequenced lambda-phage DNA digest, we see that there's only a tiny fraction of shorter reads, as we saw here, and a really dominant peak at the expected size of the full-length genome. I have to say here that this is rather a qualitative way at looking at your data than a fully quantitative assay. This correction actually is useful not only for AAVs, but for any nanopore sequencing experiments where you care about the length of your reads. And I can only recommend everyone to try it and test it. After this, however, I now want to come really at AAV-specific NGS analysis. So we really want to go now in the direction where our deep expertise with AAVs meets the deep data provided by Oxford Nanopore sequencing. And the question that is really driving us is always, we want to prepare for what may be asked tomorrow. What may be the question of a regulator in future? So can we already today establish something that will tell us even more than is asked at the moment? And the first thing is that we have, to put it in a Western reference, 3 types of AAVs: good ones, bad ones, and ugly ones. A good vector is the desired payload flanked by ITRs. This is what we want. A bad vector is a distinct undesired sequence without ITRs at the end. And the ugly ones, so the really bad ones, are those that are chimeric. So they have a distinct undesired DNA sequence, but it's fused with 1 or 2 ITRs. Why do we care so much about ITRs? Well, if we look at the fate of an AAV vector after entering the cell, there is an endosomal escape, a nuclear entry, an uncoating, so that basically the genome gets freed. And then we need the ITRs. They're absolutely required for second strand synthesis to prime it and for episome formation. And we have the formation of this long-lived episome and really long-term gene expression for months, years, maybe decades. If we have a vector payload without ITR, there's also nuclear entry and uncoating, but we cannot have second strand synthesis now. So this means we might have a transient transcription, if at all, but sooner or later, DNA degradation. We don't have this persistent long-term gene expression. And this brings us back to the beginning. Right at the beginning, the question of the FDA was, well, what is happening with the large T antigen from SV40 that has oncogenic potential in your cells? Well, if this is only transiently transcribed, probably it's not too bad. But if this can really become recombined with an ITR form and episome and have really long-term expression, it might be really a problem. To now find sequences that are fused to ITRs is really bioinformatically not a trivial task, because I don't know the sequence. For example, if it's fused to the human genome sequence, I don't know to which part of the human genome it's fused. I don't know the fusing point, so it's very difficult for me. There is no possibility to find a reference that I can map to. And always, if you miss a reference, a pipeline needs to be more complex than if I can simply map to a known reference, like, for example, a plasmid. So how did we actually tackle this question? And you see here as an example a host cell DNA ITR pipeline, but it is actually useful to detect any rare fusion event that you might have. You have the sequencing reads, you map the reads to the vector genome. And then comes the crucial part, because every mapper actually clips parts that do not align to the reference. And we now extract these non-mapped, these clipped parts and map them to the human genome. And by this we find human genome ITR fusion reads. We have depicted here only the core pipeline for this fusion detection, and there are some other steps to polish it, but in general these are the really crucial steps. And as I said, comparable strategies can be applied to detect any fusion event, which is a great plus. And another great plus is actually this pipeline is agnostic to read length. So it works also with short reads and long reads. However, if you take short reads, a big disadvantage is that they actually underrepresent GC-rich sequences. And what is the most GC-rich sequence that we can think of? Well, it's ITRs. So ITRs are strongly underrepresented in these data. So therefore, we usually rely on Oxford Nanopore sequencing, where we don't need to care at all about the GC content. It will be sequenced no matter what GC content is inside. And now let's look at real data that are actually coming out of this pipeline. And this time we took a batch really to the maximum. We have 4.5 million reads to even detect these super rare events. And you see here that, well, how many of these fusion events do we detect? Very, very, very few. 0.092%, so less than 0.1% of all reads are host cell DNA ITR fusion events. And if we take a deeper dive, you see here that a lot of them are rather ITR scaffolds with tiny pieces of human cell DNA inside. And only very few have ORFs of only 400 nucleotide, which would still be a tiny gene present in them. And as I said, this pipeline can be easily adapted to any other fusion event. And we also checked for rep ITR fusion, so these replication genes that are necessary for the vectors to multiply. They are actually rather genome toxic because they, for example, make DNA breaks. So we don't want to have too many of them in our batch. And if you look here, they are even rarer, less than 0.03%. And if you look now, how many of them have only a single full-length rep gene, it's 79 out of 4.5 million, or 0.0017%. And this, once again, is really a nice testimony to the high quality of our EPIC system and the high quality of rAAV vectors that comes out of it. With this, I'm at the end of my part of the presentation, and I will hand it back to Rebecca, who will wrap up this presentation and give you a summary.
Thanks, Florian, for giving a good overview on our pipelines. I just want to now use the opportunity to summarize what we've just heard. And in short, we wanted to show you that we have developed a robust long-read NGS platform based on Oxford Nanopore sequencing for deep AAV characterization. And beyond our standard pipeline, we have added specialized workflows, for example, the impurity linkage analysis, impurity ITR linkage analysis that Flo just presented to you to further de-risk AAV products. Generally, our platform can be expanded into other manufacturing platforms. So again, we do offer high flexibility, not only in terms of the plasmid system, but also in terms of the manufacturing platform itself. So if you're working with insect cells or with other modalities, I'm sure we can accommodate that. And we can maybe even expand it into other virus modalities. And this flexibility in general ensures that we stay ahead of our current and future requirements. So if you are now interested, I just want to give you a bit of logistics and timeline. As we've mentioned in our presentation today, we only need very little amount, and we can even start with development batches of lower yield. So we only need 1 e to the 11 vector genomes in total for a full analysis, and we would need all potential reference sequences from your end. We then typically process your data in around 15 business days and report back to you in a meeting. We do target around 500,000 reads per sample to give you a good statistical overview on all impurities as well. And as I've mentioned before, we will have a meeting to report everything to you, to explain everything to you, so you will get our expert analysis, as well as a report that can be in a standard or an advanced format. As mentioned before, we will always adapt our pipelines to your requirements. And if you have more questions, you can reach out to our email address in the bottom here. And all the questions that you will write to this email address will be forwarded to our NGS team, consisting of the 3 people that you have met today. If you're interested in our recent publications or abstracts, you can find them on our website. And there is another hint to our publication that summarizes all the results in a lot more detail. And with that, I would like to thank you for your attention, and I'm pretty happy to now start, or that we can now start the Q&A session, and we are excited for your questions.